So, like I said, Friday we finished up with eukaryotic or with bacterial gene expression. Today we are going to focus on eukaryotic gene expression. Last week we talked about like negative and positive gene expression or gene control, and we got more in depth or detailed with the systems at play. We talked about specific operons, all that fun stuff. Eukaryotic gene expression is a little bit more complicated. So in order to kind of get through it and work through it, we're not going to talk about specifics. We're going to go through a more generalization, because instead of just like one or two ways to control gene expression, eukaryotes have a bunch. Tomorrow, we are going to expand on one of the things that we are going to talk about today. We might have a little one of the article breakdowns, if I can find a good article for y'all. And then we will work through a couple more eukaryotic gene regulation things. We will finish up with cell specialization and cancer with the cell cycle control stuff on Friday. Next week, we are going to wrap up our gene expression unit. So we're aiming for some sort of assessment next Wednesday, Thursday, with Monday, Tuesday focused on reviewing. And then if we are good to go after our review Tuesday, we will do our assessment. If we need some more time, we can push it a day, and we will start our next stuff the week after. So, nothing too crazy. Any questions? All right. So, today is all about the many stages of eukaryotic gene regulation and how they are controlled. So, gene regulation in eukaryotes can be controlled at many different stages. The first one we are going to talk about is structural gene regulation. Before we get that, we have to figure out why we need so much gene regulation in eukaryotes. So, bacteria are one cell. Yes, they don't have different cells needing different information, carrying out different tasks. Their cells aren't specialized. We have a bunch of different cells with a bunch of different tasks. Our heart cells do something different than our skin cells. Our skin cells do something different than our kidney cells. Our kidney cells do something different than our brain cells. So, while all the cells in an individual, nearly all, some of them have a little bit extra in there. But almost all the cells in our body have the same genome. The DNA in your toes is the same as the DNA in your eyes. And because of that, only about 20% of the protein coding sequences in a specific cell in our bodies is expressed at any given time. So we have to have a way to control which of the genome will be expressed in which cell and when. So, the differences in cell types aren't due to differences in genes, but due to differential gene expression. And that's kind of the whole goal of today, is to show all of the different ways that eukaryotes use to make sure that each cell in their body does what it is supposed to do. Which is why cell specialization is a big part of this week. So, just because the cells on your scalp look and feel different than the cells in your blood, it doesn't mean that they have different DNA. It just means that they express different genes. With bacteria, we had we controlled expression through transcription, right? Stopping transcription, starting transcription, increasing the rate of transcription, decreasing the rate of transcription. And yes, that is something that humans do too. That is something that eukaryotes do too. But we also have a lot of different levels at which this control takes place. So we're going to talk about structural chromatin control. We're going to talk about transcriptional control. We're going to talk about translational control, and we're going to talk about control on the protein level. Yes. Usually, our bodies are really good at saying, "Oh, that cell isn't doing what it's supposed to be doing," and then it's programmed to apoptosis. So, if it's a mutation in a part of the DNA that isn't expressed, it could just let lie, and we wouldn't know about it, and you can't pass that down to the next generation because it's not in your germline. But if it's something important, usually cells are really good at saying, okay, that's bad, cut it off. When that doesn't happen, that's when we can get cancer, which is what we're going to talk about on Friday. Yeah. Mm-hmm. Yeah. Same DNA, yes, but that protein, that hemoglobin protein, isn't going to the disruption in that hemoglobin protein isn't going to mess up anything else. That makes sense. Yeah. Well, that segment of DNA just codes for hemoglobin. So if the cell doesn't use or make hemoglobin, then the mutation is not going to affect that cell at all. Good question. So yeah, starting us off, we are going to cover structural gene regulation, or ways that chromatin and chromosome structure can affect what genes are transcribed or what genes are accessible by certain proteins. Starting us off is the regulation of chromosome structure. So, the location of a gene's promoter relative to the placement of the proteins that the DNA is wrapped around, and the sites where DNA attaches to chromosome scaffolds affect whether or not a gene is transcribed. So, we remember chromatin structure is DNA wrapped around histone proteins, wrapped around each other, wrapped around each other, wrapped around each other, and then condensed into a chromosome. If a gene is hard to access, if a gene is hard to get to, if it's in the center of that mess, it's going to be very, very hard to transcribe. Or if when the DNA is unwound, right, it's still around that chromatin structure. Even if it's not condensed into chromosomes, it's still got the chromosome proteins laying around, and they're still loosely attached to them. If certain segments of DNA are more tightly packed than others, they're harder to get to. If they're harder to get to, you don't transcribe them. So, we talked about Barr bodies and how people with two X chromosomes, one of them is always deactivated and all wrapped up. That's a type of chromosome structure regulation. If we don't need it, wrap it up, put it away. We can't access it. If proteins can't or if we can't access it, we can't transcribe it, and therefore those genes are not expressed. So that's one way. Another way is through histone modification. The DNA is wrapped around histone proteins, and histone proteins are proteins, so they have an end to that polypeptide chain. That end of the polypeptide chain sticks out of that protein. And when it sticks out of that protein kind of bundle, it would be, where's my protein thing? It'd be like if I had my little protein guy and it had a tail sticking out like this, right? If the DNA is wrapped around this is still accessible to enzymes. So there is a process called histone acetylation, where we can add special, specifically acetyl groups, to the last amino acid in that polypeptide chain. This acetylation of histone proteins separates histones. They can't get close enough because those acetyl groups will kind of bump into one another. This relaxes the chromatin, relaxes our DNA, and lets transcription happen more easily because the DNA is more accessible. As you can see in this picture right here, normal DNA chromatin complexes, we've got histones that are very tightly wrapped around one or tightly connected to one another. We've got DNA wrapped around. If we add those acetyl groups, we have a bigger space between those histone proteins, and we can increase transcription that way. So if you're making a nerve cell, right, and you want to easily access the enzymes needed for nerve cell things, right, we might get histone acetylation on those chromosomes in order to access those genes in a easier, faster way. It's like you would be more likely to read a book if it was open in front of you than if it were locked up in a cabinet. Yes. Histone proteins that DNA is wrapped around. DNA is on the histone proteins. It just like if this is our histone protein and this is our DNA molecule, DNA will wrap itself around it just to keep it from tangling, and then it'll find another histone protein, wrap around it, and then all of these will wrap around one another and coil up like my phone cord, and that is chromatin. So if we had a bunch of these red proteins really, really close to one another, it'd be harder to access this DNA. But if there was space between them, and the space was particularly a promoter, it'd be way easier for transcription to occur. We can also have proteins that have the opposite effect, and that comes with DNA methylation. There is another special set of enzymes that can methylate DNA itself, that can actually make changes to DNA rather than the histone proteins, where they add methyl groups to certain bases of the DNA. This is usually cytosine. And when DNA is methylated, it is more tightly packed together or more tightly bound to the histone protein. When DNA is methylated, when DNA is methylated, it can't be transcribed. Those methyl groups don't let RNA polymerase bind properly. It cannot be transcribed. So again, if we've got genes we don't need, so so our skin cells don't need to know how to make insulin, right? That's our pancreas's job. So the gene to make insulin might be methylated in the cells that don't need to make insulin. They wrap it up. We don't need it. So we don't even have to worry about wasting resources. So we've got chromatin structure, we've got histone acetylation, we've got DNA methylation. All three are ways that we can change DNA structure or chromatin structure to affect what can be transcribed and when it can be transcribed. Some of these processes can also be reversible, right? Sometimes our bodies don't need access to stuff, like prepubescent people don't need access to all of the genes related to puberty, and they could probably be undone when they need them. All of these changes all of these changes can be grouped into a special study called epigenetics. So, these modifications, acetylation, methylation, don't change the DNA sequence. Don't change the DNA sequence. But they can be passed along to future generations of cells. If a certain segment of DNA is methylated in one part of, or in a cell, it can pass those methyl groups to the next generation. This inheritance of traits transmitted by mechanisms that aren't nucleotide sequence is called epigenetic inheritance, and that's what we're going to talk about tomorrow. This can the study of epigenetics is, we believe, part of the reason that if we have identical twins, one can inherit or one can get or exhibit symptoms of a genetic disease while others don't, while the other one doesn't, specifically because of inheriting not the sequence, but how the sequence is structured. So we're going to talk about that tomorrow. Questions about epigenetics? When you think about chromosome structure in bacteria versus eukaryotes, right? We can understand why this chromatin structure helps regulate transcription in eukaryotes, but not bacteria, right? Bacteria don't have as much DNA as eukaryotes do. Bacteria have circular chromosomes where everything is accessible all the time. We have a lot of DNA, so we store it differently, which allows us to control it at the structural level. But we can also control gene expression through transcription, specifically transcription initiation, getting that first process started, just like bacteria do, right? But we have more complicated ways of doing it because we are more complicated individuals. So, one way we can do it is through organization. So, this is our typical gene structure down here, and they have multiple control elements or segments of non-coding DNA that serve as binding sites. That should be sites, not sires, for the proteins called transcription factors, which bind to control elements and regulate transcription. So it's a little bit wordy. Essentially, we have a lot of non-coding DNA segments called control elements that act as binding sites for proteins called transcription factors. Transcription factors are any proteins that alter or affect the rate or ability of transcription. So in our picture right here, we have our promoter where we start. We have our untranslated region that helps us bind to the ribosome. We have our open reading frame, our start and stop codon. We have that segment of RNA after our stop codon. And then we have these other things here. We have enhancers, silencers, promoters. Those are all of our regulatory sequences. They are our control elements that help us change the way we transcribe things. So we're going to go through some specific examples of these control regions and transcription factors. So we have general transcription factors and we have specific transcription factors. General transcription factors are essential for the transcription of all protein coding sequences. So if there is something that is involved in the transcription of all proteins, we call that a general transcription factor. The big example here are our TATA-related proteins. There are proteins that bind to the TATA box in our DNA sequence that help our RNA polymerase bind. That TATA box is in all protein coding sequences, all DNA coding sequences. Therefore, the proteins associated with it are considered general transcription factors. Now, not everything is controlled by the same set of systems. Some have more, some have less, and this is where specific transcription factors come into play. High levels of transcription genes at the appropriate time and place depend on control elements and a bunch of specific proteins. These are considered specific transcription factors. So thinking back to our trip operon, right? We had a special trip repressor. That trip repressor only represses the tryptophan operon. Therefore, it is a specific transcription factor. Whereas our TATA binding proteins exclusively bind to the TATA box, which is involved in all transcription. So it is a general transcription factor. How are we doing so far? I think this is the wordiest slide I have. So, enhancers are a type of control element. An enhancer is a group of control elements that will be thousands, or could be thousands of nucleotides upstream or downstream of a gene, or it even could be within an intron. The whole point of an enhancer is to help stabilize our transcription initiation complex. A gene can have multiple enhancers that are activated in different scenarios. And enhancers are typically associated with only one gene and no others. So in this example right here, we've got an enhancer that is a pretty decent ways away upstream from our promoter. The enhancer can bind activators, which help bind our transcription factors that allow our RNA polymerase to bind to the promoter, stabilizing RNA polymerase and helping it get started and get going. So if this enhancer relies on the structure of our DNA molecule and how it is a kind of linear thing. So being able to fold that DNA over and bind that transcription factor group helps RNA polymerase grab on and move forward. So enhancers can be here. You could even have an enhancer down here that also, bends forward or bends backward or just associates with transcription factors in some way, shape, or form. So they can be in introns, they can be way upstream, they can be way downstream. If a transcription factor is over here, it could loop around this way and bind. Enhancers are groups of control elements that are not immediately associated with the promoter. These are all transcriptional regulation methods. We also have the combination control of gene expression. So, on average, enhancers can be up to 10 control elements. Each control element can bind with one or two specific transcription factors. So there are a lot of transcription factors that can be involved with these enhancers. And the combination of control elements in an enhancer is very, very important to that transcription rather than just the fact that the enhancer exists. So if some but not all of the requirements for a certain gene to be transcribed is available, it might just be transcribed at a lower level. So everything kind of just depends on how many of the control elements within that enhancer are bound or activated. So if only about half of them are bound, or if only one or two of them are bound, RNA polymerase might be strong enough to grab on and start transcribing, but it also might not be. We also have coordinately controlled genes, which is what we talked about with our operons, our trip operon, our lac operon, which are genes that are clustered into an operon and regulated by a single promoter and transcribed into a single molecule. So we've got multiple genes all at once. That way we don't have to worry about transcription factors for each one of those genes, right? Our lac digestion, or not our, in E. coli, their lac digestion depended on 3 different enzymes. Instead of having to turn on all 3 of those enzymes with individual promoters, we were able to clump them together. We do the same thing. We can also clump genes together to help make 1 or 2 specific molecules for a specific purpose. So if a protein is made up of 2 polypeptides, like this one I have in my hand right here, if a protein is made up of 2 polypeptides, instead of having each of those polypeptides be transcribed separately and translated separately, we can loop them together so we don't have to worry about controlling each individual portion. We can do them both together. Those were all pre-transcriptional things about how transcription happens. There are also ways we can regulate gene expression after transcription occurs and before we've translated officially. So, post-transcriptional regulation, one of the things we can do is through RNA processing. We talked about introns and exons. What's really cool about this is that we have this process called alternative splicing. In alternative splicing, different mRNA molecules are produced from the same primary transcript, and that depends on which segments are treated as introns and which segments are treated as exons. So this bit right here, and we've got our example, our pre-mRNA is red, yellow, green, blue. This first example, the yellow is an exon and the green is an intron. Introns go in the trash, exons are expressed. But and it creates a protein with 3 distinct shapes or 3 distinct polypeptides, right? This mRNA sequence treated the yellow like an intron and the green like an exon. So we have similar protein shapes, but not identical. Remember, protein shape determines function. So even changing the shape just a little bit can really affect what the outcome is. So if we had 2 proteins, right, they both are receptor proteins, right? We'll say we have 2 receptor proteins. One of them is signaled by one thing, one of them is signaled by a different thing, but the way they release that signal into the cell is the same, right? They both trigger the same thing, but they have different activators. The only thing that needs to change between those 2 proteins is the active site itself. So I would be code for the dispensing mechanism twice when we can just change that one aspect of that activation site. So if this right here, if the star and the little blue oval are parts of the protein that latch onto the membrane or release a signaling molecule, and the yellow and the green bits are the different active sites, right? We wouldn't want to have this red and blue segment be transcribed twice. We want to be efficient. So we can slice up the mRNA differently and get different proteins, which is really stinking cool. So that's mRNA processing. Another way we can do it is through the initiation of translation, right? If the mRNA can't get into the ribosome, it will not be translated, therefore it will not be expressed. So for some mRNAs, the initiation of translation can be blocked by certain proteins that bind to sequences or structures within the UTRs. So if you remember, our mRNAs have untranslated regions before the start codon and after the stop codon. Those regions can bind to special proteins in the cytoplasm and block that mRNA from entering a ribosome. So if we are triggered to make a protein, right, and then immediately we realize we do not need that protein, but we've already made the mRNA, we can stop that mRNA from entering the ribosome by binding a certain sequence on that untranslated region. So it blocks gene expression in a way that doesn't use transcription. It blocks gene expression by blocking translation. We also have mRNA degradation. So in bacteria, or in general, I guess, the lifespan of mRNA in the cytoplasm is very important in determining how often protein synthesis happens, right? So if the mRNA sticks around for a while, it can be translated a bunch of times. But if the mRNA falls apart after it is used, we're not going to translate it anymore. In bacteria, they have special enzymes that go through and they munch the mRNA after it's being translated because bacteria need to adapt to changing environments quickly. We have mRNA lifespans that are kind of controlled or influenced by these UTRs in the beginning of that mRNA. This can mean that mRNA lasts for hours, days, or even weeks, because usually conditions in our bodies don't change as quickly as they do for a bacteria. So our mRNA can stick around longer and be used for longer. So the amount of time that the mRNA are allowed to exist within the cytoplasm also affects the gene expression of that specific protein. The last one is protein processing and degradation. So we can also control how often proteins are expressed by how long those proteins are allowed to exist in the cell. So polypeptides uh, are typically processed to become fully functional proteins, right? There's that section of RNA or of polypeptide folding, making sure it has the right structure, being targeted for where it needs to go. In these steps, we can regulate where they go and how often they go there. So in these regulatory steps that process polypeptides, or we can regulate, goodness gracious, in these modifying and transporting steps of polypeptide processing, we can take regulatory action to control where they go, how they get there, and how often they get there. We can also very strictly regulate the length of time proteins function in a cell. So we have selective degradation, meaning we can choose which protein we can degrade and when. So if you have an enzyme that digests we'll just use lactose as an example. If you have an enzyme in your body that digests lactose, right? If lactose is no longer around anymore, we don't need those lactase enzymes. We can munch those up so we don't waste energy. And we can reuse the pieces. A lot of times when we break down proteins, right, we can use those amino acids to build more proteins. If we break down mRNA, we can use those nucleotides to make more RNA. Perfect. Got 5 minutes left. There is an exit ticket on Candace, or they will be in about 2 minutes. So if you can knock that out for